welcome to BioTown. So today we're going to talk about a very interesting topic, and at the same time we're going to start a new chapter, and the chapter name is Microbes in Human Health and Welfare. So as we all know, these microbes are really tiny organisms, but they cannot be seen, viewed under the naked eye, right? So these tiny organisms, they can be viewed only under the microscope, but when you actually enlarge them, actually when you magnify them, these microorganisms have a lot of good things which make humans' life better, right? So that is what this chapter is all about. We're going to study the various uses of microorganisms. So with this motivation, if you didn't subscribe our channel, please do subscribe and press the bell icon for further updates, and we'll move on to the chapter. So microbes. So as we all know, these microbes can be measured only with the help of microns. So microns are a sort of symbol with which you can measure, right? It's like millimeter, centimeter, meter, kilometer, the same way. So the same way, these microorganisms are abundant in number. So we have bacteria, viruses, fungi, algae, we have prions, so n number of bacteria, the microbes. So this chapter deals with the important microbes along with their uses, right? So now the microbes, you can actually divide them into two categories. One is for personal use. Personal use is something where the household items, how one particular household item is converted into the other elegant form with the help of these microbes. And the other thing is industrial products, like commercial products, where we make in the beer industry, wine industry, or in the medical industry, where they manufacture various enzymes and they manufacture vaccines and all these things, right? So that we divide the microbes, the uses of microbes, into two different categories. One category is based on the personal use, the household, and the other is commercial. So first we will talk about the household microbes. So the first thing is we have Lactobacillus acidophilus, Lactobacillus lactis, Streptobacillus lactis, and Streptobacillus thermophilus, Lactobacillus bulgaricus. What you all feel like this is Greek and Latin? Yes, obviously these words are derived from, not exactly from English origin, they are all from Greek and Latin. So now these set of microorganisms, they are all called as lab, which means lactic acid bacteria. So the major work of these microbes is to live in high acidic environment. Maybe you can take lesser than 6 pH, the potentiality of hydrogen lesser than 6. So these microorganisms, they dwell in environment where the pH is lower, right, than the neutral level. So that's why we call it as lactic acid bacillus. But why do we name them lactic acid? It's because they help in, supposing there's a milk, they convert lactose in the milk into lactic acid. So any substances, they make use of the substances and they convert the substance into lactic acid. So that's why we call it as lactic acid bacteria or lactic acid producing bacteria. That's all, right? So now bacteria itself is a plural form, bacterium is a singular form, so make sure you don't use it bacterias, right? There's no such word. So yes, so first, for example, I will give you a very common example. So your mom is buying milk, right? And the milk, she's heated up the milk, and now she's allowed the milk to be in a warmer phase. Now what happens? She does something called as inoculation process. So biologically we call it as inoculation process, but basically what is inoculation? Adding certain amount of bacteria into something. So the same way, when a mom wants to make curd, what does she do? She takes milk, she heats up the milk and allows the milk to cool or become warmer, right? So the warm milk, what does she do? She take a spoon of the starter culture. Starter culture meaning the curd, the curd, leftover curd, the yesterday's curd she takes up, she mixes in the milk and she never disturbs the milk and allows it to cool, right? So that's what she does. But your mom is a great scientist. It's because she does something like what all the lab technicians or researchers could do. That's all, friends. It's very simple. So basically, the way she adds the curd, previous day curd, is called as starter culture, and she mixing it up right with the milk, and that is called as inoculum or inoculation procedure. So this is what basically this is the basis of a high-tech, hi-fi laboratory. So these lactic acid bacilli, bacteria, what do they do? Suppose there's a milk, they convert the lactose into lactic acid. So now we have curd, yogurt, cheese, paneer. Now what is the difference between these different forms of milk is that nothing. It is all based on the inoculation you add. For example, when we want to convert milk into curd, all these three important bacteria, they take up the load. Lactobacillus acidophilus, lactobacillus lactis, and streptobacillus lactis. So now all these three bacteria which is there in the starter culture has been mixed with milk, right? Now what do they do? In the milk, we have a protein component called as casein. Casein. Now this casein is there, no? These microorganisms, they start breaking down these casein. So now as a result, what will happen? The milk will start coagulating. So the milk will start becoming thicker. So that's how we get curd. So wait for 24 hours or 12 hours. So night she puts and early morning she wakes up, she keep it in the refrigerator. So that's all curd over. So this is a basic phenomena where we deal about the lactobacillus bacteria. Next, in the formation of yogurt. So yogurts, we have even more thicker texture than we comparing it with curd, right? So it is all because of the other two bacteria. One is Streptococcus thermophilus and Lactobacillus bulgaricus. Now what do these bacteria do is that in the milk, they convert the lactose into lactic acid. So that's why we get a sort of sourness in yogurt, right? And also they add little of flavors, right? Strawberry flavored, vanilla flavor yogurt. So that is little commercialization, right? Because to enhance the flavor, and that's why, and only then we go and buy it from the market. So it is because of that. But basically microbe play a very important role. And then cheese, paneer, if it is cheese, you have this Propionibacterium, Shermani, and all these different types of bacterium where n number of carbon dioxide is also being liberated as a byproduct. Why? It's because cheese have holes in them, right? So that's basically because of that. And also based on the flavor, we have American cheese, cottage cheese, the different types of cheese. So based on the flavor of the cheese, the bacteria also they vary. So this is all about the uses of microbes in household products. Along with that, idli batter, dosa batter. So all that, what does mom do? When there's an idli batter, she takes up the dough and she starts mixing up with her hand. So now what exactly happens? The microbes, they play a very important role. She mixes it up, aerates it. So while she's mixing it up, the process is called as aeration, which means that she is directly or indirectly, she is passing air in the dough, right, in the batter. So that is basically to deal with idli and dosa batter. So now we are done with microbes in household products. Next is SCP. So SCP, it stands for single cell proteins. Now why are they called as single cell proteins? It's all because these are various microorganisms. They're just unicellular. They just have single cell, but collectively when you pile them up, they are much rich in nutrients. So when you want to build up your body, or when there is any deficiency of vitamins, proteins, calcium, so we basically have these SCPs. So SCPs varies. So one such example is spirulina, right? And now microbes in industrial products. So this is basically commercialization, not done in household. So first is antibiotic production. So we have various antibiotics, right? Penicillin is obtained from Penicillium chrysogenum or Penicillium notatum. It is all because they produce such byproducts. So it could be either a primary metabolite or a secondary metabolite, but basically they're all metabolites, right? So So then antibiotic resistance. So what do we do in biotechnology? We inject the gene of interest and then we ligate it and then we apply restriction enzymes and then we clone it to specific vector, the plasmid, right? So all this we would have learned earlier. So this is because this is the antibiotic resistant process. And then fermentation. So fermentation is a basic principle of making idli batter or dosa batter, whatever. So what will happen when air is passed along with the microbial activity? The dough raises, the batter raises, right? So it is that is fermentation, and only because of the process of fermentation you can actually produce this antibiotic production, antibiotic resistance, and all that. Next is chemical and enzymes. So chemicals and enzymes are produced by these microorganisms either in a primary metabolite form or secondary metabolite form. Next is sewage treatment. So we have a lot of Coom water. So there's a lot of research going on how to convert this Coom water into some usable form of water, at least for toiletry purposes. So it is because, and this is because of sewage treatment, where we collect all this bad water consisting of all the microbes and bad chemicals, and then with the help of the same bacteria or the fungi or algae, we convert the sewage water into a fresh water, at least if not into a drinkable form, but still a potable water, usable water. Next is biogas. So biogas, this is emerging research field where instead of our LPG gases, is there any possibility of using gases which has been produced as a result of microbial activity? And then we have biofertilization, biopest and all we have, which means that when you use all these components, for example, if it is a cotton and there is a cotton borer worm which actually infects the cotton, when you use sort of fertilizer or sort of biosprays, what exactly happens? These bacteria or the fungi will never allow these worms to infect the plant. And then we have bioremediation. So imagine it's a huge ocean and there is an oil spill nearly in the center part of the ocean. So what do we do? Nothing could be done, right? So rather we introduce our superheroes, we call it as superbugs. We have Pseudomonas, where when you just spread these Pseudomonas, they actually utilize the metabolites and they produce the other metabolites, secondary metabolites forms. So that's it all about this part. So we spoke about the microbes in household products and then microbes in industrial products. In the forthcoming parts, so I'm going to actually describe each of these in depth. Thank you.